Hey, what's up, guys? So, in class, what we discussed previously here was how proteins are made, right? So, again, from this whole seminar, we started looking at how the DNA molecule is structured, what it looks like, and then how that structure plays into its function, right? And so today in class, we had identified how we went from a DNA molecule to then an RNA strand, which then gives the code to produce a protein. And then those proteins inevitably make us everything that we are, and also provide a lot of the major functions that we have in our bodies. So the next step that we're going to kind of jump into is this idea of genetic engineering and what that means and the significance of it and what possibilities does it have in our future as a society. So when we talk about this idea of genetic engineering, the biggest thing to kind of wrap your brain around is that it's when you look at your DNA, right, we have the code of bases. It's going into those codes and changing them up a little, whether you're adding in things, whether you're taking things out or altering the code that is there. Inevitably, what that's doing is it's changing the protein that you make from that particular gene. And so this has been messed with, this has been experimented with, and we've used this a lot in today's world. And there's unlimited possibility in the future of how this can be used. So today I want to kind of jump into this a little bit and give you some background, as this is where we're going to go next week. This is really kind of what we're going to jump into and wrap this seminar up with, is how do we use DNA in current times? Now that we know about it, now that we know what it looks like and how it works, how can we use it to improve the life of living organisms? So I want you to start by looking at these two mice. Okay, so obviously the glaring difference between these two groups is the ones on the right are glowing, right? They're glow in the dark. Okay, and that is due to genetic engineering. So these particular mice on the right, they have had a gene manipulated or a gene changed to where the protein that it produces is a glow-in-the-dark, fluorescent, glowing protein. And so that is one way that we've experimented with this that you can see right in front of you. So to kind of jump into what this is, what genetic engineering is. It is the direct modification of an organism's genome. It's a direct change in an organism's genes, right? And that gene is a specific code for a specific protein. And so when you change those things, it changes the protein, which, as you'll see here, changing the genome enables engineers to give desirable properties. So we can choose what characteristics or what things are this organism going to show by manipulating their DNA. Okay, you've actually heard about this a lot. This term right here, genetically modified organisms. Anything, any living organism that has been modified, has been genetically engineered, that's what we call a GMO. And you see this all over the place in the supermarket. You see non-GMO foods. You hear about GMOs. All that is an organism that has been modified. So just to kind of give you a little background, this has been in the process for a while as far as how we can use this. Back to the 70s is where this all kind of really started, which if you kind of think back to where we looked as far as the history of DNA, it's about 15, 20 years after we discovered what DNA really looks like that we started to be able to manipulate it. And so that started back in 73 when we created the first genetically modified bacteria. Okay, and that bacteria, then we used that technology from that bacteria to then create some genetically modified mice. So obviously stepping up into larger organisms. Then we started in 82, the first commercial development of GMOs. This was a bacteria that produces insulin, which is what's necessary to allow sugars into your cell. And so this was produced for increasing in insulin production that we can then use in people with diabetes so that they can have insulin. And so we grew that through bacteria. 94, we began to sell genetically modified food. So a lot of your produce has been genetically modified. And then in 2003, we began to sell GMOs as pets, right? So there's glowfish, little fish like this that are glow in the dark. They're fluorescent, just like we saw in those mice. And so as you see, we've progressively gotten more and more involved in this process of GMOs. And so what does that look like? There's many different ways that this looks like, but inevitably, the genetic changes that we're making in a GMO affects the protein synthesis, the creation of the protein, which is what we talked about in class today. So by changing which proteins are produced, we can affect the overall traits of the organism, because again, we talked about proteins being the thing that makes everything that we are, that makes us up, and that makes all the characteristics that we see. And so genetic modifications can be completed by a number of different methods. You can insert new genetic material randomly or in targeted spaces. So if you had a strand that was AAA and you wanted to change that, you could put in a TTT in the middle of that, and that would change the protein that's being made. You can directly replace genes. So if let's say I have the short gene and I want to put in the tall gene, I can take the short gene out, put in the tall gene, and then look at me, I'm not short anymore, right? Or you can remove genes altogether. And then another way here is you can mutate, you can manipulate or change up the letters within existing genes, again, with the whole idea being that it changes the protein that it produces. So GMO bacteria, just a couple examples here that I want to give you. So GMO bacteria, bacterias are the most common GMOs because they're super simple. Okay, we can really easily manipulate them because they're really simple. The reason why we haven't really done a whole lot with humans, couple reasons. Big reason, we're really not simple. We're pretty complex. And so one of the most interesting uses for genetically modified bacteria is the production of hydrocarbons, plastics, and fuels, which usually only found in fossil fuels. Okay, so we use these bacteria to produce different things. If you look here, there's two examples. E. coli is a bacteria that we've actually modified to produce diesel fuel, okay, which is kind of crazy to think about. But when you're talking about fossil fuels, things that aren't unlimited, we can produce that same product by using a living thing in bacteria. Another way that we see GMOs is plants. So consider this when we talk about plants and how we can genetically engineer them. What problems can they solve? What problems do we have in the world that can be solved by modifying the way that these plants work? So as you'll see here, it says consider world hunger. Can we feed people better? Can we grow produce in a desert with minimal water? Can we produce a crop that if we genetically modify it, it can grow in the harshest of places? That would change the way that we provide food throughout the world. Weather problems. You have insecticides, pollution, right? Instead of spraying all your crops with poison, we can manipulate the DNA to actually prevent insects from eating them. So couple examples here. Okay, so GMOs when it comes to crops. The first that we really did this was in the 2000s, and what you see here, the GMO crop production in the U.S. back in 2010 was 93% of soybeans, 93% of cotton, 86% of corn, and 95% of sugar beets. So think about it. All of these things are common foods, common produce, and we've taken these and we've genetically modified them to allow them to grow better, to withstand harsher conditions, to be more plentiful. There's many different ways in which we can modify these. So the one example that I gave here, which I gave earlier, was BT corn, which is a type of corn where a gene from the BT bacteria is added to the corn, which produces a protein that is poisonous to certain insects, but it's not poisonous to humans. That's huge, right? You keep insects away, but people can still eat it. A couple other examples here: banana vaccines. We've modified viruses, or we've modified virus injected into a tree, causing bananas to contain virus proteins, which then can allow you to obviously create a vaccine. Venomous cabbage. Scorpion genes have been added to cabbage to prevent insects from eating it. Okay, the same thing that we saw with the corn. So other reasons why we might do this: insect resistance, herbicide resistance, drought, freeze, harsh conditions, disease resistance, to produce more, to produce it faster, to increase the nutrition, and then also last longer. So animals, little different, okay, but we still can obviously do this in animals, like we talked about. We can add, or we can manipulate genes to be glow in the dark, right? We did that with mice, as you'll see here. Glowfish, we've also done that as well. Fast-growing salmon. We have manipulated genes in salmon to allow them to grow faster. Why is that important? Well, we can make a lot of money off of it if we can sell a big fish, right? And then cows here. We can modify bacteria that are responsible for methane production in cattle, which makes them less smelly. Cows release a lot of methane into the air through their farts, and we can actually manipulate that bacteria to smell better. So what I want us to consider is this, and this is where we're going to go. Could Spider-Man be real? Right? Can we do this in people? Well, what we're starting to see is that we're growing this process, and it's going to continue to grow. What we've actually done is, within goats, we've produced or genetically modified the genes in goats. We've taken spider genes and put them into the goats to allow them to produce spider silk in the goat milk. And so the question to be had is, where does this go? And how far can we take it? And can we use this in people to manipulate the way people look? Can we have a book in front of us and say, I want my child to look like this, and then modify it to do so? So those are realistic possibilities, but there's some big time questions. When we consider GMOs, we have to consider, what's the risk to human health? Are they unsafe? Specifically with the crops, are they unsafe to eat? Okay, we also have to consider harm to the environment and wildlife. One example I gave in class was growing trees taller and faster, right? If we grow that tree can still reproduce and give its seed to another tree. And then you end up having these massive, fast-growing trees in neighborhoods and inevitably getting too tall to maybe possibly falling down or just overcoming a particular area that it shouldn't. Increased pesticide or herbicidal use, right? We have to be careful of using those things. Farmers' health, okay? Again, they're using these things, they're poisonous to certain insects. At what point is it too much? And is there things that we don't know as far as how they can infect the health of the individual using these things? The seed and pollen drift, that's the whole idea with the trees. And then can you create these things and then inevitably they evolve into resisting what you made them to be? But then the whole human idea is what we really want to think about. Nearly 50 countries around the world, including Australia, Japan, and all of the countries in the European Union, have enacted specific restrictions or full bans on the production and sale of GMOs. And the reason being, are they safe? We don't know. We don't know enough at this point to really discern whether they're safe to use or not. But as we go, we're going to start considering, how can these be used in people? How can we use them in living things to make life